Hey there, and welcome. Today we're going to dive into something that literally powers our entire planet. It happens all around us, in plain sight, but it holds some amazing secrets. We're unlocking the science of photosynthesis. So, have you ever been hanging out by a pond on a sunny day and really looked closely at the plants underwater? You might have seen these little streams of tiny bubbles rising up. It's one of those things you might not even notice, right? But what you're looking at is, well, it's basically magic. Science magic, of course. Yeah? Those little bubbles? They're a clue. They're the visible proof of this incredible chemical reaction that's happening right in front of you. You can almost think of it as the plant, breathing out. And that gas? Well, that's literally the breath of life for Earth. I mean, it's kind of wild to think about, right? Those tiny bubbles hold the key to a process that fuels, well, pretty much every ecosystem, creates the food on our plates, and makes the very air we're breathing right now. All right, so let's get right down to it. What exactly is this secret recipe that plants have totally mastered? So at its heart, photosynthesis is just how plants make their own lunch. They take really simple ingredients from their surroundings, use sunlight as the energy source, and turn it all into chemical energy, a type of sugar called glucose. And that leftover stuff, the byproduct, that's the oxygen we were just talking about popping up in those bubbles. So the recipe itself looks super simple, right? A plant sucks up water, breathes in carbon dioxide from the air, adds a little blast of light energy from the sun, and bam, it cooks up glucose to power itself, and as a bonus for us, it releases oxygen. Okay, so knowing the recipe is one thing, but how do scientists actually, study this? How can we see it in action and actually measure what's going on? Let's head into the lab. This is the big question, right? If you're a biologist, you've got to find a way to measure how fast this whole thing is happening. And you know what? Those little bubbles we saw before, they are the perfect tool. And this setup? Oh, it's a classic for a reason. It's just brilliant. You've got a lamp playing the part of the sun, giving off light energy. You've got your pondweed sitting in water, which has carbon dioxide dissolved in it. The pondweed gets to work, and we can figure out how hard it's working just by counting the bubbles. Simple as that. So the actual method is super straightforward. You put your lamp at a set distance, let's say 10 centimeters away, and for exactly 1 minute, you literally just count every bubble that floats up from the plant. Then you move the lamp back to 20 centimeters. Do it again, then 30 centimeters, and so on. By changing the distance, we're changing the intensity of the light hitting the plant. Now, for this to be actual science, it has to be a fair test. You know the drill. That means we can only change one thing at a time. In this case, the light intensity. Everything else, what scientists call the controlled variables, has to stay exactly the same. The water temperature, how much carbon dioxide is available, even using the exact same piece of pondweed. All of that is absolutely crucial to get results you can trust. Okay, so we've done the experiment, we've got our numbers, we've counted our bubbles. Now for the fun part. Let's see what story the data is telling us. So check this out. Here's some data from an experiment just like the one we've been talking about. You can see the pattern right away. With a pretty low light intensity of 25, we get 19 bubbles a minute. But when we crank that light all the way up to 100, the rate skyrockets to 222 bubbles. I mean, the trend is pretty obvious, isn't it? More light equals way more photosynthesis. Well, up to a point anyway. But numbers in a table are one thing. When we actually plot this on a graph, the story really pops. You can see it so much more clearly. Just look at the shape of this thing. At the beginning, as we turn up the light, the rate of photosynthesis just shoots up in a nice straight line. They're what we call directly proportional. Double the light, you get double the bubbles. But then, something happens. The line starts to curve and eventually just flattens out into a plateau. And that plateau, that flat part, that's where things get really interesting. It introduces us to one of the most important ideas in biology, the concept of a limiting factor. So that's the big puzzle, right? We're giving the plant all this extra light, all this energy, but at a certain point, it just stops going any faster. Why? What's holding it back? The answer is a concept called a limiting factor. The best way to think about it is like a factory assembly line. You can have the fastest workers in the world, that's our light energy, but if you run out of parts to assemble, that's our carbon dioxide, the whole line grinds to a halt. It doesn't matter how fast your workers are, they're limited by the supply of parts. It's the same for the plant. Even with tons of light, if it runs short on CO2 or if the temperature isn't right, the process hits a ceiling. And this is a perfect illustration of how different things can become that bottleneck. On the left, you've got our light intensity graph with that classic plateau. Now look over at the temperature graph on the right. The rate goes up and up, hits a sweet spot, and then it just crashes. Why? Because if things get too hot, the special proteins, the enzymes, that make photosynthesis happen start to break down. They lose their shape, a process called denaturation. Basically, they just fall apart and stop working. Two different factors, two totally different kinds of limits. All right, so we've seen how this all works in a nice, clean, controlled lab setting. But what about out there, in the real world, in a messy place like a lake? How do these ideas hold up? So what we're looking at here is a graph showing the population of algae, those are tiny little photosynthetic life forms, in a real lake in the UK over a whole year. And the pattern is just impossible to miss. The population is low in the winter, it starts to take off in the spring, it goes crazy in the summer, and then it drops back down in the fall. So what's behind this massive seasonal swing? Why do the algae just go wild in the summer? By now, the answer should be starting to click. You guessed it. It all comes back to those limiting factors we talked about. Summer is the perfect storm for algae. You've got longer days, which means way more light intensity, and the water is warmer, which brings the temperature up closer to that optimal point. With both the light and temperature limits lifted, the algae can photosynthesize like crazy. And the result? A huge population explosion. So think about the journey we just took. We went from a single bubble in a tiny test tube all the way to the seasonal cycle of an entire lake. And it's all explained by the same fundamental idea of limiting factors. And it leaves us with one final and much, much bigger question to chew on. We now know that light, temperature, and carbon dioxide are the main dials that control this life-giving process for the whole planet. So what happens when we start turning those dials on a global scale? That's definitely something to think about.